I'm going to start talking, and I'm going to assume that maybe this will start functioning. So the talk is Never Say Never Lyse Again. And the second slide I'll show is where that title comes from, which is a James Bond movie. I'm quite fond of the Renault R5 turbos. They're such a wonderful JC. And so what I'm going to be talking about is something that I call physiological lysis delay. So I've been interested in lysis inhibition for a very long time. My first paper was on lysis inhibition and why it exists. It's a phenomenon where phage like T4 affects a cell like E. coli, and then if a second phage comes along and absorbs, not affects, but absorbs to the same cell, it stimulates the first infection to extend itself actually for hours and make much larger burst slices. And the idea was that this phenomenon is a way of adapting physiologically to high cell concentrations where there's also lots of phages around, so there aren't a lot of new cells to infect. So at that time you want to change what you do and your way you're infecting, so that instead of getting in and out of the cells quickly, you're instead of getting in the cells taking maximum advantage of what is there for the virus to exploit and make lots of phages. Now that I've extended and extended, so I have this argument that that viruses like T4 use this phenomenon while they're infecting biofilms in order to produce large numbers of virions, so those virions can go off and not die in the process, but maybe find new biofilms to infect. And the more virions that you produce, the higher the likelihood that some of them will survive and find new bacteria to infect some distance from the original biofilm. Alternatively, when you're exploiting within a biofilm, especially if you have competition, it pays to be able to get in and out of cells quickly so you can go from cell to cell to cell. And you can imagine that the virus will go from cell to cell to cell. Some of those cells will be lysis inhibited and make lots of new viruses. They'll go off and maybe at least one of those viruses, phages, will find a new biofilm to infect and start the process all over again. So somewhere towards the end of this talk, I talk about there being 5 lytic strategies. So one of them is the normal, ordinary infection where the virus goes in and makes some virions, it lyses after, in the case of T4, maybe 20-25 minutes. That you can describe as rapid lysis. Rapid lysis actually was originally a phenomenon that was used to describe lysis inhibited defective virus cells, phages, and I just use rapid lysis to describe the normal infection process. Lysis inhibition is the extended infection process. At the other end of the spectrum, if a phage is infecting a stationary phage cell, they might not make any viruses at all, but if you put them into an environment where there are more nutrients, the lytic infection might restart and they'll make virions. Alternatively, they might go into the stationary phage cells and make a few virions, but not a very large burst size at all. So what I'm hoping to actually show is that it's been known for quite some time that people are doing virulence assays, or something I call a coat assay, which I'll talk about slightly if I get any visuals, where you see flashes of light, where if you vary the starting titer of the virus and you follow the infection via optical density, then what happens is that at the lower starting titers, the bacteria are able to grow more before the phages catch up with the bacteria and infect all the bacteria. And you can actually, to some degree, be able to actually quantify what the starting titer was based upon what the curve looks like. And if you start with a lower enough concentration of viruses, then you'll end up with the phages catching up with the bacteria as the bacteria reach relatively high concentrations. And at that point, it looks like the phages displaying a very long latent period. Those latent periods actually can last for hours. And with phage T5, which is what I focused on, I can get these infections to last for 8 hours. And then you see something, in fact, the lysis starts relatively early, but it just seems to go on and go on and go on. And what I have done is an experiment where I actually keep track of the amount of virions that are produced. I do it by lysing cells artificially using chloroform. And over time, you watch these T5 infected cells that are going on for 8 hours, and they're producing new virions the whole time. You get these wonderful straight line curves. And so the idea seems to be that these are initiated at the 0 point, the infections are maybe at a 0.88 an hour or two, and they just keep going and going and going, making virions. Now what seems to be happening is that you have these very long infections that are associated with bacteria that are not as physiologically healthy as we're used to working with in the laboratory. We tend to use mid-log phase bacteria, or we tend to use stationary phase bacteria, but there's not a lot of focus on the middle. And I accidentally started doing this because I wasn't doing experiments for a while, and I went back in, and I grew up my cells too much, and I was like, everything changed in how these were behaving. And so that seems to be what's going on. So I have a way of explaining this whole phenomenon in terms of microcolonies. Now it was nice to see Stephen's microcolonies, because I'm like, look, microcolonies, they're relevant to what I'm going to talk about. So if you think of a microcolony, clonal microcolony, it starts with a single cell. They expand via cell replication. The cells in the center are starved of nutrients and oxygen, so they are not growing anymore. The cells on the outside can be growing very rapidly, more rapidly than log phase in raw culture. And then somewhere in the middle, you have presumably late log phase cells. These are cells that don't have access to so much nutrient and oxygen. So when a phage comes along and infects a microcolony, the first thing it's going to do is go through this rapid lysis, go in, make some phages, come out again. Now, if there's secondary absorption after these, let me start again. If after these infections lyse and they start infecting other cells on the same microcolony, if some of those cells are absorbed twice or more, then they can display lysis inhibition and ultimately end up in larger burst sizes. But as these phages start penetrating into the microcolonies, they start encountering cells that are in later log phase, and what they seem to be able to do, at least with T5 and T3, T6, I mean, all of them seem to display these very long latent periods when infecting these cells that are higher density and they're presumably worse off physiology for the phages. They seem to be able to display these long infections. And they're presumably doing it because their object is to make lots of virions so that they can go off and in terms of biofilms and maybe find another cell to infect another biofilm and get started. Now, T5 displays these long infection periods, but it also can't infect cells that are of a relatively high optical density and therefore cell density. But T1 and T3 and T7 seem to be able to infect cells and still display longer latent periods, but at least eventually lyse. When you have a phage that displays lysis inhibition, T6 is my favorite one just because it's so crazy. So T6 actually will give me 24-hour plus infections that actually eventually lyse. Now, I haven't gone in there and made sure that they're actually producing virions after that point, but they actually display these curves that eventually they turn into lysis. The reason I haven't done that with the T5 experiment, I'm in the lab do that yet. But that's basically my idea. The idea is that these phages, at least the ones that I work with, if you raise the concentration of bacteria enough, they have these very long latent periods. That's been shown before, but I don't know how much people have been paying attention to it. But they eventually can lyse if we let things go long enough. And with T5 at least, I've been able to show that if I artificially lyse these bacteria, that we have over an 8-hour span of time a constant increase in the amount of phages that have been produced. And I guess we're not going to see my talk, but that's essentially all that I wanted to say. People have shown this before, and I've moved the bar a little bit further along, and I have a theory as to why it's relevant, especially for biofilms. I've got a little bit of funding from the NIH to do the experiments. I mean, I'm working on like $5,000 to buy equipment to get things together, which allowed me to do these experiments. Actually, I should point out, so I've been trying to figure out who is the person who did the first automated 96-well plate lysis profile. So this is using a kinetic microtiter plate reader. And so far, I have traced it all the way back to Addison et al., which is a Betty Cutter paper, so very relevant here. I'm trying to figure out if anybody has done an older one than, I guess an older one than that. What was that?
What year was it?
That was 1998. It was in genetics, Addison et al. And the interesting thing is that the person who did those experiments was me. So it's possible that I started the whole process of using microtiter plate readers to do lysis profiles. So I want to thank Kasia Danis Borczyk. Everybody knows her as Katarzyna, as her film name. For getting me started on looking at lysis inhibition in focus that led to these things. And also Vivek, I don't remember his last name. What's Vivek's last name? Okay, thank you. For doing the sequencing, so I actually know that T1 and T5 are actually those correct phages. The T1 turns out to be a contaminant in my lab. I never brought it into my lab, but apparently 5 or so of my phage stops are T1. And I think that's more or less everything that I intended